Oh, hey. Let's start. Oh, yes. Okay. I guess that's probably fine. These folks are going to get like a very serious reading. You can just bring it back here. How far does the cable reach?
Not that far, actually.
Okay, maybe we can. Okay. I mean, yeah, that's fine. Okay. Okay, I can also just enter in the URL.
Oh, okay.
Because yeah, it's just send it to you.
So before starting the event, we have some wine and drinks here over there. So please feel free to take.
Yeah, so I don't look like a lush being the only one drinking this beer. Enormous beer.
That's what it's all about. Going to tell you when you're fat.
I'll bring tequila. Yeah. Oh, okay, yeah. Thanks. Was it more dry?
Thank you.
It's an ice cream. Oh, whoa. Then I realized
How's the volume? Good.
Good. Yeah, really good.
Yeah, I know.
No, I didn't. But I did go to the run by the government thing.
Okay. What is your Google?
I know. A lot of them is no, not yet. You have to.
Okay,
Go.
what's your
Is
email?
it in the script? Yes, it is.
It's gonna be busy. Okay, I'm so excited. They'll be like,
Stop being an influencer.
I'm gonna wake up earlier than today.
It starts.
Does it? I can have a bit. I feel like my taste
Oh my gosh, I'm going to publish my content
interesting stuff. I'm just gonna read it.
Yeah.
You're right. I should send it. Oh,
Yeah. One person.
You're a funny film guy. That PowerPoint. Yeah. You always have to have or I just see the students have yeah. Well, it's communicative. Rather go to yes. I've already been there, so that's fine.
Okay, cool.
Yep.
Okay.
Oh, yeah, I should start this. you can have your.
Thank you. I'll give you a little more tipsy while you.
Yeah. So thank you everyone for coming to the talk tonight, and it's my great pleasure to introduce you Danbee, my great friend and also my colleague in college. And also, it's also my pleasure to introduce Danbee to our lovely Good Enough family. Yeah, so a brief introduction about Danbee. Danbee went to school in the US with a degree in brain and cognitive science. We just reviewed that. So I don't need a cheat sheet. And then she moved to Portugal for her PhD programs in PhD training in neuroscience. But the whole lab moved to London a few years ago. So part of her PhD research is done in the UK. And this year is your final years of PhD, so I guess I should wish you a successful year. Yeah, and I met Danbee when I first moved to London, so she is in fact one of the first person I met in London. Yeah, and although we have only worked for a few months, she has been a great inspiration in my scientific career. Yeah, especially I think one of the great lessons I learned from her is that the first step in helping people is to get to know them. So that means even though we are still students with limited resources, we can still do something, and the whole world will respond to that. And when looking back, I think one thing I inspire her a lot is that we don't really have to compromise our principle for the sake of climbing the career ladder. So I hope you have a wonderful night, and let's welcome Danbee.
Very nice introduction. Yeah, thank you everyone for being here, and for thank you to Chiu for inviting me to come give a talk. It's actually pretty convenient because, as Chiu mentioned, I'm in the final throes of the PhD, and so this is actually a nice chance for me to practice my defense, kind of, sort of. Probably won't be as casual. I probably won't be drinking a beer. Okay, so the title of the port talk is How to Dance Like a Cuttlefish, and yes, you will learn how to dance like a cuttlefish. And in parentheses, I put the official title that I'm putting on my thesis document, which is On the Aims and Methods of Field Neuroscience. And so I'm gonna tell you a little bit about what this means and why I think field neuroscience is necessary. So this is just a quick overview of what I'm going to be talking about. I'll first explain to you what I mean by field neuroscience and why I think it's an important aspect of neuroscience research going forward. And then I will talk about the three main projects that I have been working on during my PhD. So, okay, why field neuroscience? So I like to go back in history to when modern neuroscience was sort of first birthed. And it surprised me, and it has surprised, I guess, consequently, a lot of other people I've told, that neuroscience as we know of it today didn't really exist as a thing, as a discipline called neuroscience until the late 1900s, specifically in the 50s, 60s, and then in the 70s. This is when the first annual review of neuroscience was published, and in it, W.M. Cohen, the head editor, he remarked on the emergence of this new interdisciplinary field called neuroscience, and specifically, he phrased the goal of neuroscience as trying to understand how the nervous system controls behavior. And this felt really important to me to realize this and to go back and find this reference, because sort of throughout the whole course of my PhD, I felt that myself and also a lot of my colleagues and the people around me, there was sort of this identity crisis happening in neuroscience where people were like, what the hell is neuroscience? What are we supposed to be doing? There's so many subfields of neuroscience now, and people put neuro in front of everything, like neuroeconomics, ethics, neuro law. There's just what the hell is neuroscience? And so it was really refreshing to go back and see historically, why did people even want to have this term neuroscience in the first place? And so I like taking that as my starting point. What people wanted to do, the reason why people wanted to band together and become a field called neuroscience was to understand how the nervous system controls behavior. So, okay, so it's like, okay, we have this assumption that that's what nervous systems do. They control behavior, and now we just have to figure out what it is. So then it was also interesting to, again, going back to the history of neuroscience as a field, to observe how people went about this, and also the questioning people did of, actually, are we sure that what nervous systems do is control behavior? So before the 1960s, 70s, when people thought about, okay, what is it that nervous systems do? People used to think, okay, they do something that we call cognition. And cognition is all the invisible stuff that you do, like thinking, meditating, all these things that are inside your head that nobody sees. And so it's this invisible thing. And so that obviously made it quite challenging to understand what the nervous system is doing. And then there was a lot more like anatomical work, and our tools and techniques improved. And in the 1990s, there was this newer idea and a new phrase that came about called embodied cognition. And this idea is the idea that the way that you think is deeply embedded in the body that you have, and sort of the situation that you live in, and the environments in which you develop, and the relationships that you have. And so it's sort of, it's in your body, but also it's deeply embedded in what your body is doing. And so then this developed even further. So now there's this new term that I also learned quite recently called psychomotricity, which is this idea that it's actually a completely closed feedback loop, that the way that you think is influenced by the way that you act, and the way that you act is influenced by the way that you think. One of my favorite science fiction authors, Ursula K. Le Guin, does a lot of writing about how language is inherently tied to the ways that we think, but also the ways that we think are manifested in the ways that we speak and the vocabulary that we use. And so this is becoming sort of this growing sort of side trend. It's not necessarily the mainstream, like, biggest, most popular theory in neuroscience, but this is sort of a growing side trend that people are acknowledging, that the bodies that nervous systems are in and the environments that nervous systems interact with, they have a huge impact on what we call cognition, what nervous systems do. So sort of to I don't know, exemplify this, one of my favorite examples is, so I'm a big marine animal lover, and there's this marine animal called the sea squirt. This is the like official scientific name, but I like sea squirt. So sea squirts have a very interesting life cycle, where at the beginning of their lives, they're this little larva. They look like this, and they have like a tail that rotates around, and they move through the ocean, hunting food and looking for the perfect home in which to settle. And when they are in this larval stage, they have a very developed nervous system, and it's quite complex, and it runs throughout their whole body. You can see labeled the notochord, which is similar to a spine in any vertebrate. But then once this sea squirt has found the perfect piece of coral or the perfect rock, it settles down, it changes its shape, and it digests 90% of its nervous system. It eats it. Because our nervous systems are actually very, they're really high maintenance. It requires a lot of energy to maintain a nervous system. So some of us just had dinner. The vast majority of what we just ate is going to go towards feeding our nervous system. It's the hungriest organ system in our body. And so now it's presumably, it doesn't need a nervous system anymore, so that's why it ate it. And so now it's stationary, and it does have a very rudimentary nervous system. People are in debate about this because it's literally just one nerve, and it controls the movement of pulling in water in order to collect nutrients and food, and then shoving things back out again. So this animal, to me, sort of exemplifies how, if you don't need it, why bother having such a costly organ system? When do you need it? When you're moving. The only time you need it is when you're moving. And so this led me to this idea about what can we learn about nervous systems by carefully observing the movements of bodies. If what a nervous system is doing is controlling behavior, and if we break that down even further, we could say basically a nervous system is there in order to coordinate and mediate all of the millions of cells that we're made out of so that we can move as a single fluid being, then maybe this is another way that we can observe and deeply learn about nervous systems without having to surgically cut into bodies, which would also alter what you're doing. This is sort of a deeper methodological debate that I have often, which is if in order to get at the nervous system, you're cutting into the body and you're cutting into the nervous system. I mean, I know that when I have a pin stuck in my body, I move differently because I think, oh gosh, there's something here and it kind of hurts and it's kind of delicate. And so my whole behavior changes. Everything that I do changes, and even the way that I think changes. So that must apply in some way to the animals that we study in the lab. So how can we kind of work around that? Is there a way to work around that? And so that's sort of where field neuroscience comes in, this idea that I want to study freely behaving organisms, nervous systems that are not being held captive, that are not under pain of surgery. Although, just to be clear, I do think there's a lot of value to invasive techniques inside of a laboratory. I think there's a lot of things that we are learning, and that's also very important. But if that's the only thing we're learning, I think we're missing a big piece of it. And some people recently published a paper saying that they also agree that neuroscience needs more behavior, that as a field, we've kind of gotten into this rut of we only study these very highly reduced, highly simplified situations inside of laboratories, and we're losing sight of this incredible, rich variety of behaviors that exist in the real world, and we just have no handle on how to study this. And again, in this paper, what they assert, and which I agree with, which is why I'm quoting them, is that understanding behavior and its component processes at the level of detail necessary to generate meaningful neural level insights will require an emphasis on natural behaviors performed by individuals. So this is where I thought, okay, so I want to start developing more tools and techniques for observing nervous systems non-invasively in natural settings. So that is how I'm defining field neuroscience, non-invasive techniques for observing nervous systems in natural settings. So brief tangent. So while I was doing my PhD, and as maybe some of you may also be able to sympathize with this, I ran into a lot of changes to my life situation, health problems, mental health concerns. There were a lot of challenges to doing a PhD that were not, covered by my interactions with my advisor or the mentors and thesis committees and all these things that are sort of provided as structure by academia. And I ended up getting a lot of help from some friends of mine who were in a band. And I realized that making music was a really important way for me to maintain my mental health and my ability to be at the level that I needed to be in order to do PhD research. And so I ended up writing a small collection of songs about my PhD, and I'm going to share them with you along the way during this talk. And so this is the first song. It's called Mind Shape. And I just put the lyrics up there because they kind of have to, I don't know, they do reflect, I think, aspects of my research, and so I didn't want them to get lost, but all right. When our minds begin to shape, faced an unknown landscape full of fatal fruits, but we move 'cause that's what living things do. Yeah, we groove and we rise to unchain our minds from the vines of the vices of the status quo. Oh. will labor for the myth of a rich man's favor. Instead of loving their neighbor, we all long to belong, sing along harmonies of all the same song. Oh, 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 tell you about the first major project that I did for my PhD, which was a set of experiments with cuttlefish called the Cuttle Shuttle. And the reason why I started with cuttlefish, well, okay, so before I started my PhD, I was working at the Museum of Science in Boston. And I was a presenter for the current science and technology department. And what we do is we would find stories in the news about current science and technology. And we would develop these presentations that we would do on the museum floor. And it was like a 3-week cycle, 1 week of preparing the presentation, 2 weeks of actually giving the presentation. And during that last week, we were preparing the next one. So it was a really fast, exciting cycle of stories. And 1 week we picked a story about how the US military was investing a lot of money into studying cuttlefish because they use active camouflage, and they're supposed to be, I guess, not overloading the computer's ability to play this very incredible GIF image of a cuttlefish changing its shape, its color, its texture. So active camouflage is different from passive camouflage in that it happens very rapidly. And in cuttlefish and cephalopods, so this is a family of marine animals that includes squid and octopus, the way that they change their the color and the texture of their skin is they have these tiny structures that are acted upon by muscles, that are controlled by nerves, where the nerve body is in their central nervous system. So it's literally controlled by their brain. So it's very fast. They can change within milliseconds, and some of them can even do this like crazy strobing thing where they're just constantly changing colors. Go look it up on YouTube. I guess this slide is just not happening. So yeah, they. So I found these creatures and I found them incredibly fascinating. And after I finished my bachelor's degree, I was really burnt out with academia, and I said, I'm never going back to school. There's no way I'm going to go to grad school. And then while I was working on this story, I was like, oh shit, I just really want to spend all my time studying cuttlefish. Wait, who's going to give me a job to just let me study some weird, obscure animal all the time with no possible application or, profit to come out of it? Oh wait, that's what academia is. So then I was like, okay, well then maybe I should go to grad school and see if I can, study neuroscience with cuttlefish. Some other interesting points about cuttlefish is they have these W-shaped pupils. So this is their eye. And their pupils dilate, they grow and shrink just like ours, except ours is a circle and it grows and shrinks as a circle. Theirs is a W and it grows and shrinks as a W, which is absolutely wild. But the eyeball itself is very, very similar to the human eye. So this is one of the many examples by which cuttlefish and cephalopods are this amazing comparative example of a creature whose their biology is organized in a completely different way. They evolved in a completely different environment. And yet, in the end, they've converged on some solutions. For instance, how do you decode light? They've converged on a solution that is very, very similar to humans. Oh yeah, so this is my little animated GIF of humans, we are sensitive to the frequency of light, and this is how fast the light is bouncing. And so different colors are bouncing at different frequencies. But cuttlefish are actually, so this is why they have the W-shaped pupil, this was only discovered recently, they are sensitive to polarization, which is the direction that the light is traveling when it bounces off of objects. So they can't really see colors, but they're able to see polarization. And we also have some information now that suggests that polarization might be a secret communication channel for them, because even though they live in the ocean, a lot of their predators are visual in a way that is more similar to us. They're sensitive to frequency, but cuttlefish can see on the polarization of light, and they have structures in their skin that allow them to very delicately control exactly the polarization of light as it bounces off their bodies. So now there's all this speculation that they have a secret communication channel through polarization. Very exciting. Yeah, so I was like, okay, I really, really want to study cuttlefish. I want to do neuroscience with cuttlefish, but there are, at the time, no neuroscience labs that were set up to do any neuroscience with cuttlefish. So my advisor was like, all right, fine, let's just find like the best marine biologist in the world who's studying cuttlefish and offer to do a neuroscience collaboration with them. And hilariously enough, they were on Cape Cod in Massachusetts, where I had just left. I left Boston thinking, I can't handle any more Boston winters. I'm going to go to Lisbon for their nice weather. And I got sent right back in order to do my science. So anyways, I ended up at the Marine Biological Laboratory at Woods Hole on Cape Cod. And I worked with these two lovely humans, Kendra and Roger. And so my advisor's lab, most of the people there work with rats. And one of the main topics that we study are, you know, how do intelligent systems come about? What does it mean to be intelligent? Well, okay, so maybe it means that you're able to predict things really well. You create a great model of the world in your mind such that you can predict things and simulate things really well. Okay, well, what if there are other agents in the world that are also predicting things, and now you have to predict agents that are also able to predict you? So then we kind of converged on this, okay, hunting. Hunting is this behavior that animals do. It's very ethologically relevant. That's the term, fancy term that scientists use for like, oh, it's really important in the wild. But it's basically an act of trying to out-predict each other, right? So like, I'm hunting something, I think you're gonna go there, so I'm gonna try to outsmart you and do something that you can't predict so that I can eventually catch you and eat you, or not be caught and not be eaten. So I was like, hey, I would like to study prey capture in cuttlefish. Is there anything interesting going on there with their skin when they do this? And Roger, he's been studying cuttlefish for like his entire career, and he was like, yeah, actually, so there's this thing that like marine biologists and scuba divers, they notice that after a cuttlefish releases its tentacles while it's hunting, they have this weird like color burp. And it doesn't make any sense to us because it makes them really visually conspicuous, but presumably this is a moment when they're very vulnerable, they just caught food, maybe they're distracted. Why the hell, an animal that lives, survives by hiding, why would you make yourself more visible in this moment when you're presumably very vulnerable and very distracted? So everyone's really confused about this. And so I was like, great, perfect, I'll study that. So then I was like, okay, how do I create a hunting scenario with cuttlefish? So I decided to build a robotic prey, which is fancy talk for, I put a little bit of shrimp at the end of a plastic skewer that I hot glue gunned to like a hobby motor that was controlled by an Arduino somewhere. And I put a GoPro camera under the water. And this is like the hunting box. The hunting box was inside of this bigger tank, a holding tank, mostly because I didn't want to have to bother with making my hunting box waterproof. So I'll just stick it inside of something else that's already waterproof. We have a overhead camera here. We got water into the hunting box with these tiny little tubes so that we could create as minimal surface disruption as possible, because this overhead camera, you know, it's going to be, if there's a lot of surface disruption, that'll cause lots of difficulties. There were still difficulties later with analysis. So yeah, this was the setup, and as you can see, my very sophisticated robotic prey over here. And then once we got an animal in there, what we had to do was convince it that this is something that it can eat. So what we did was, so at Woods Hole, during the summers, we can just go out to the harbor and there's like these little glass shrimp that we can just kind of harvest, and we put them into the home tubs and they hunt it and they love it. It's their natural prey. But in the winter, there's no shrimp in the harbor. So what we do is we go to the grocery store and we get cocktail shrimp, defrost it and cut it into little chunks, put it on the end of these skewers and wave it in front of them and be like, no, seriously, this is alive, eat it. So I basically replicated that with my robotic prey. And luckily, enough of the cuttlefish were convinced that I got lots of video of them hunting, and I was using a high-speed camera, and so I was able to get high-speed video of this moment immediately after they throw their tentacles. Okay, so this is pretty well described in the literature from all the way back in like the 1920s. They already knew about these 3 phases. They position themselves and then watch the tentacles, they shoot out. And then this thing, it's like their back becomes like a chessboard that like you zoomed out suddenly and there's like lots of tiny little chess blocks. So then now we're gonna take a closer look. We've zoomed in and I have painstakingly edited every frame so that the cuttlefish body stays in the exact same spot. We slowed down the video. So now you're seeing the tentacles slowly pushing out. And then at some point they go ballistic. So it's like, da, 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 kapow. And then the tentacle shot pattern appears. And then just to prove that it's not just this one time and this one animal, well, actually, okay, next I'm gonna show you all of the tentacle shots of this animal. So this is all of the times that it shot its tentacles and it caught something. Now this is all one animal, but you notice how many different patterns it's wearing. And then, so it's gonna go, tentacles go ballistic, and now all of them, every moment has this weird color burp. Really weird, right? Okay, so then I was like, okay, well, what about when it misses? Okay, so this is same animal. These are all the times it shot its tentacles and it didn't catch the prey. So watch closely again. Tentacles go ballistic. And you kind of see it, but then it disappears. Like, where did it go? So, you know, sometimes it does stay, and it's like when it gets stuck on the side of the hunting box, or like its tentacles got stuck on the plastic skewer, and it didn't actually get to eat anything. So, you know, it's not pleased. So yeah, like I have, you know, several theories about what might be going on. Here we go. Okay, so then here's some plots and maths and statistics to appease my thesis committee. Yeah, so basically I pooled across all of my animals and I said every single time that there's a catch versus every single time there's a miss, how long does this weird, you know, color pattern persist? And oh gosh, the projector doesn't do favors to this graph. So the blue line is all the catches, and the red line is all the misses. Basically, about half a second after the tentacles go ballistic, after they are shot, there is a statistically significant divergence in the edginess of the cuttlefish's body pattern. So I tried a bunch of different ways of like measuring this body pattern. So like, as you guys saw, as a human watching this video, it's like, oh yeah, there's definitely something going on. I can see something happening. And when it's missed, it kind of happens, but then it goes away. But then I was like, okay, I need a way to describe this numerically. A, because it would be really convenient if I could teach a computer how to find this moment. But B, like, the idea would be I need to mathematically describe this so that it's unambiguous. It's not just me saying, oh, subjectively, I feel like this is something, you know, distinct happening. I want to be able to say, yes, mathematically, there is something distinct happening. So we tried like looking at luminance values. I even like mapped luminance to audio and then like put it into the frequency range that we hear in. And there's YouTube videos on this. I'm happy to share afterwards. But anyways, the only measure that seemed to really, really like distinguish between catches and misses was there's this algorithm called canny edge detector. It's a machine learning algorithm that's supposed to look for high contrast edges in an image. And so as you saw, that checkerboard pattern, there was a lot of like blocks, tiny blocks of black and white. So it suddenly got very edgy. And I love being able to say, I'm studying how how edgy cuttlefish are. So that was basically the main finding. And I don't know why this thing won't go away, but we have a paper that's going to be posted on bioRxiv soon with links to all the videos. So yay! So now we're going to get to our next song, which I'm going to require some audience participation. And actually, Chiyu, could I get your help with this? Chiyu's going to help by being our lead dancer. So this is the part where you're all learning how to dance like a cuttlefish. Okay. So as you see, there's two verses and there's also a chorus. There is already a dance for the first verse and the chorus. There is no official dance for the second verse, but please feel free to improvise your own. So this song encapsulates basically everything that I have learned about cuttlefish during my PhD. So let's start with verse 1. So W eyes, we learned earlier that cuttlefish have W-shaped pupils. So everyone's going to hold up 3 fingers. These are your cuttlefish eyes, and they are usually on the sides of their heads, okay? So they're on the sides of your head, and you're looking around, you're just chilling out, I'm a cuttlefish. And then when you want to hunt, so what cuttlefish do is, because their bodies are squishy, what they do is they actually squish their face and bring their eyes forward so that the vision from both eyeballs converge, and they have better depth and motion perception for when they're hunting. So they squish to the front when the shrimp's on the run. Okay, then you're gonna morph your hands from being your eyes to being your cuttlefish arms and tentacles. So cuttlefish have 8 arms, so these are your 4 fingers, and they have 2 tentacles, which are your thumbs. And when they're just chilling, hanging out in the ocean, your tentacles are tucked away, hidden behind your arms, and your arms are just kind of doing this. Yeah, they're just right here, here. I'm just a cuttlefish hanging out in the ocean. Okay, all right, so a Morpheus tangle got 8 arms to wrangle. Then they have no spines, so wiggle your spine because you've got one, but they don't. But they are masterminds of fooling your sight. Excellent. Okay, so let's do just the verse. Let's do just the verse together. All right, ready? W eyes looking side to side, squish to the front when the shrimp's on the run. Now, a Morpheus tangle got 8 arms to wrangle. No spine, but masterminds of fooling your side. Excellent. Well done, everyone. Okay. All right. Do you guys think you can handle one more dance for the chorus? Okay. So the chorus is about their hunting behavior. Okay. So now, when cuttlefish are in hunting mode, what they do is, okay, go back to being, I'm just a cuttlefish hanging out in the ocean. Yep. When you notice food, the way that you indicate this is your 2 middle arms are going to go up. Yes. So are you ready to hunt? You're all going to say yes. Then middle arms up. Then you're going to sneak, stretch. This is because they are stealth hunters. They're not pursuit hunters. They're not cheetahs or lions or wolves. They get into like a bit of sand and they kind of wiggle in under the sand and then they wait really patiently for something to come close, okay? Then, now prepare the tentacles. This one's a little tricky, but I totally have faith in you. So, who's ever done the awkward llama head? Yes? Okay, so make a llama. And now what you're going to do is you're going to go back to it being your cuttlefish arms, because what they do is they make this weird tripod where, so these, the middle arms are up, the next 4 arms create like a barrel for your tentacles, and this is what guides out the tentacles. Do you remember seeing the tentacles kind of slowly peeking out in the videos? And then the very bottom arms, they're kind of like stabilizers, okay? So they're in this weird tripod position, okay? So make a tripod and throw. So you're gonna shoot your thumbs out. Yes. Okay, great. Then I'm gonna ask you, did you catch or miss? The pattern will let you know. So what I want you to do, because unfortunately we cannot change our skin and texture, but if you have caught your prey, you're gonna dance wildly and wiggle. And if you haven't caught your prey and you're like, no, I didn't catch it this time. You're gonna go back to cuttlefish hanging out and sneak around a bit more, okay?
This
All right.
one is difficult. I didn't know this.
I wanted to make sure you were also challenged. Didn't want you to get bored. Okay, so let's do the chorus together. Ready? All right. Are you ready to hunt? Yes. Then middle arms up and sneak, 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 stretch, 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 stretch. Now prepare the tentacles. Make a tripod and throw. Did you catch or miss? The pattern will let you know. Yay! Okay. All right. Let's practice with some music. And she is going to continue demonstrating up front, just in case anyone gets lost. Okay. So the way that the song is going to go is we're going to do verse, chorus, second verse, and then chorus again. Okay? Is everyone ready? Okay. All right. All right. Excuse me, verse. W eyes looking side to side, squish to the front when the shrimp's on the run. A Morpheus tangle, got 8 arms to wrangle. No spine, but mastermind, a fool in your side. Nice. Okay. Yep. Are you ready to hunt? Yes. Then middle arms up. Sneak, 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 stretch, 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 stretch. Now prepare the tentacles. Make a tripod and throw. Did you catch or miss? The pattern will let you know. Yeah! All right. Verse 2. Chromatic array in the skin is how they betray when they decide whether to attack or hide. But when they have a secret, they know how to keep it. 'Cause like they polarize for colorblind All right. Are you ready to hunt? Yes. Then middle arms up. Sneak, 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 stretch, 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 stretch. Now prepare the tentacles. Make a tripod and throw. Did you catch or miss? Pattern will let you know. Oh yeah! Am I still good? Yes? Okay. All right. So, okay, well now you all know how to dance like Cuttlefish, so you can all get a PhD also. All right, so I'm gonna rapid fire go through the last few bits. So, okay, when we came to London, I wanted to continue doing cuttlefish experiments, and we developed this relationship with the Sea Life Centre in Brighton. And like I was going to help them with their animal husbandry, and then they were going to let me do, non-invasive behavior experiments with their cuttlefish. It was all a great plan until so we tried to get cuttlefish, and normally like fishermen around Brighton will get extra cuttlefish eggs caught onto their like lobster traps or crab traps. And that year apparently there were no cuttlefish laying eggs, and so we're like, okay. And to their credit, Sea Life Brighton was like, they tried really hard to get me cuttlefish. Like they tried mailing them in from like a lab in France, but like literally, I didn't realize this. They were just mailing like eggs in a box and sending it through the post. And so like half of them died on the way here. And then the Sea Life Center like started, they had like this problem with their like water filtration system. So they had to like shut down like half of their tanks. And so anyways, there was a lot of trouble. And so I said, okay, you know what? I wanted to do a comparative study anyway. And I wanted to look at how to do field neuroscience with humans because, I mean, like, I'm a human. I know what natural environments for humans are like. And in fact, this should be even easier than, you know, figuring out field neuroscience for other species. So we decided to basically, embed experiments inside of interactive installations. And the Sea Life Center was really helpful with this. So this is the cuttlefish tank at the Sea Life Center. This is where you would be looking at cuttlefish, and they said, look, you can take over this entire section, and you can do whatever you want with it. And so we ended up building this, the Sea Life Center in Brighton is in like an old Victorian building. It's like underground. It has like iron wrought like bits and like, you know, stone arches. So I was like, okay, we're totally going steampunk with this. So we decided to build something that looks like, you know, kind of like a steampunk, like submersible viewport. And basically, so cuttlefish, a lot of their nervous system is out in their skin, controlling their skin. But humans also have a portion of our brains, of our central nervous system, which is visible from the outside, which is our eyes. So all these sayings about eyes being windows to the soul, like there is some truth to that. If you look at how your eyes respond to damage, the immune system response is exactly the same as the response that your brain has to damage. There's lots of like molecular factors and like, you know, the chemistry of your retina is exactly the same as the chemistry of your central nervous system. So there's a lot of evidence to allow us to say your eyes are bits of your brain shoved out of your skull because your brain said, I really just need a handle on what's going on out there. So again, I worked with a group of amazing collaborators who all helped make this happen. Basically what we did was, so these folks built the outer casing, and then these two gentlemen helped me with the inside, the guts of the interactive part. So basically what we did was we put two little cameras right here. They were infrared cameras that would shine infrared light onto the eyes of whoever was looking in. Infrared, because that way we don't blind you while trying to record your eyes. And then we show you some videos of animals doing surprising things. I'll show you an example of what video people saw in just a moment. And then we record your eyes during these videos. And we were hoping to get, you know, a couple hundred people. Turns out it was very, very popular. And I think in the end, it was like 24,000 activations or something. Yeah, it was a huge data set. So initially, it had to be hosted because there was like a prototyping phase where I was like, I don't really know like how to explain this succinctly to people, so I'm just going to stand there all day. And it was also a great experience of talking to people about neuroscience. It was a great way to open that conversation with something that was familiar to everyone. Oh yes, do you have eyes? Great, I have eyes too. This is a great way to talk about nervous systems. And so then, you know, like getting to interact with these people and sort of after you said the same thing about 100 times every day, you start to realize what needs to go onto the exhibit. I also got to work with a student that was that I met through the Nuffield Research Placements Program. This is like a really great program that connects students from like low-income communities with researchers in various cities and at universities. And so my student was Rihanna. She also hosted the exhibit and got to practice talking about neuroscience to people. And oh yeah, here's the video. These are my eyes. Which is why I can show them to you. But this is an example of what people would see. So first they were asked, please don't move your head. There was a calibration sequence. You can see, you know, pupils are growing and shrinking and moving around. Video of animal doing a surprising thing. This part was different every time randomly picked. And then this last part, this video, I don't know if anyone has seen this video before. Yeah. So it's this, you think you're just coming upon like some rock and some ferns, and then suddenly an octopus appears out of there. So the idea was we wanted to deliberately surprise people. So this octopus video was sort of like, oh yeah, it's pretty surprising. Every time I show it to somebody who hasn't seen it before, everyone goes, whoa, what the hell? So then we wanted to basically use this. I feel like so many times in my PhD, I was like, I will do a prototyping thing, get like a pilot study done, and then I'll move on to the real science, and I never get past the pilot part. So this was like we're just gonna replicate some experiments done in the lab to show that this setup is a reliable way to, do rigorous neuroscience outside of the laboratory environment. We are able to prove that now. I'm in the throes of analyzing this data, but as you may guess, I haven't really gotten past that to doing anything else. But I feel like it's a really good argument for replication studies don't have to be boring. They don't have to be simple. You can just be like, I want to replicate this effect, but I want to do it with like 20,000 people to see if it actually works in the real world. And then it can be super interesting. So then after the initial pilot phase, we kind of changed the viewing port to make it a little more robust. And then basically the thing that I was saying to people every day 100 times over and over became these instructions that were nice and succinct and placed onto the exhibit so that it could be completely automated, no need to host anymore. So it ran in this automated state for 11 months. And that's when it collected over 24,000 activations. Out of that, it currently looks like about 20,000 of them is good data. So yeah, we lost 4,000, but it doesn't matter because we had 24,000 to start with. Coming soon, more data analysis on this is what I'm doing this month. Did you have to replicate studies done in the lab to the precision that we get in the lab by just doing it with a way bigger sample. So there are a lot of laboratory studies showing that, your pupils respond to light primarily, but they also respond to things we call cognitive loads, so things you can find attractive, surprising, arousing, shocking. And so then when those happen, your pupils should also dilate. And so, but if you look at these papers, it's 10 to 15 psychology students, upper class, white male American, and okay, so it's a very, very tiny slice of humanity. Whereas at the aquarium, especially during the summer months, we would get school groups from all over Europe, even from China. So if you, there's totally no way to be able to see it, but these are language buttons. We translated the entire exhibit and all the instructions into 5 different languages based on during that initial phase when I was hosting it. And I was realizing, okay, what kind of languages are coming through here? I think it was French, Italian, German, Chinese, English. Those are the 5. Yeah, so those are the 5 main ones. And so then the hypothesis was, if we are now looking across this way bigger sampling, is what we found in these laboratory studies unique to the very narrow sampling that was gotten in these laboratory studies, or can we say robustly that it does happen across all of these other demographics as well? People always ask, so I'll say now, we didn't collect any demographic data because we want to keep things totally anonymous, because it's kind of off-putting if you're going to the aquarium for a nice weekend, activity, and then someone's like, hey, can you fill out this form telling us all about your life? So we just wanted to keep it completely anonymous, but just to see, is it robust across age, across, socioeconomic status, across wherever it was in the world that you came from, visiting Brighton, and then you happened to be in the aquarium. So far, the analysis is showing that, the literature is correct. The hypothesis was, can we replicate things outside of the laboratory setting with enough precision that we can verify things that were done in the laboratory setting? And the way to overcome this is to make it engaging enough that you can engage tens of thousands of people. Yeah, thank you for your question. Sorry, I haven't been looking around to see if anyone else had questions this entire time. Okay, so this one, I don't have any plots for you yet because, as I said, I'm still doing the analysis. And then the last major project of my PhD has been, okay, so a little bit of context. So there's been sort of this, trend now of science communication being an important thing. There's a lot of, like initiatives for outreach and public engagement and whatnot. And something that me and my advisor and sort of our colleagues, that, while we were thinking about, what is intelligence, what is neuroscience, like what are we doing with our lives? One of the things that we kind of started thinking about and talking about and realizing is, unlike, say, particle physics, I would say very few people in the course of their life gain an intuition for particle physics. But neuroscience is studying something that everybody is carrying around in their bodies, that everybody is presumably using all the time. And so actually, neuroscience is studying something that a lot of people, and to some degree everyone, has some level of intuition about. If you've ever taken a moment to think, gosh, I wonder why I act this way every time my mom tells me not to do something, or, gosh, I wonder why I always get peeved when this happens, or, like if you've ever taken a moment to think about your own behavior, think about your own thought processes, that in a way is you engaging with what neuroscientists do, which is thinking about nervous systems, thinking about behavior. And so this whole idea of science communication and making scientific research accessible, I started to feel like it was even more important in neuroscience than in any other field, because we are studying something that is so intimate to every single one of you. And it's information that I definitely want to be made as accessible as possible, because for sure, people who are in marketing, people who are doing advertising, like people in the government, they are already using the research that's available out there to try, for whatever nefarious or not nefarious purposes they have. So I would like everyone in the population to also feel informed and empowered with the knowledge that we as a research community have been finding about nervous systems, so that everyone can make more informed decisions about how you interact with, say, ads or marketing or Google, things like this. And so I started doing a lot of outreach projects and trying to figure out ways of how can I make my research more accessible. And one of the things that I decided to do was to basically, it's this idea of the strange loop. Can a single brain ever truly understand how a brain works? Because that's the thing that I'm using to try and study the thing that, it's like this, is it ever possible for me to ever gain enough perspective to really truly understand what nervous systems are doing? Maybe the only way out of that strange loop is by expanding the number of brains that are involved, and maybe by, collectively thinking about this together. That's the only way for us to actually understand how brains work. And so I really want to get rid of this little bar at the bottom. Anyways, there was this guy in Italy called Valentino Breitenberg. He was a psychologist who wrote this book, Vehicles. And basically the idea was, let's imagine that there are some really simple vehicles. They have a light sensor, and the light sensor is connected to a motor, and that motor drives the wheels. And if we connect the light sensor to the motor with a positive feedback loop, then when there's more light, the wheels turn faster. And so it goes towards the light. So you could say that this vehicle likes the light. And so he goes through all, like, slowly, step by step, he goes through lots of simple behaviors, but all the way up to things like love and empathy and hate and revenge through these very, very simple vehicles. And at the very beginning of his book, he has this lovely quote, which I really like. And he says, "This is an exercise in fictional science, or science fiction, if you like that better, not for amusement, science fiction in the service of science. Or just science, if you agree that fiction is part of it, always was and always will be, as long as our brains are only minuscule fragments of the universe, much too small to hold all the facts of the world, but not too idle to speculate about them." So I feel like this quote encapsulates everything that I've just been saying about brains and loops and all this in a much more elegant way. So so in order to make this graphic novel thesis, I'm under time pressure and I can do some sketches, but I'm not really a good artist. And I was like, if I want people to be engaged in my research that I'm presenting as a graphic novel, I need some artists who are actually good at drawing. So I put out a call on the internet, on Twitter, on Facebook, collected a group of 17 artists from all around the world, spent a lot of time on Skype and on Slack talking to them about the script, about my vision, about neuroscience. And the idea of this graphic novel was to create a near future speculative fiction about what will happen to us, the planet, and to humanity, but also to us neuroscientists in the next 50 to 100 years. And, you know, it's been a little eerie because I did a bunch of research in totally unrelated fields to mine, but mostly looking for how do I think the world will play out. I ended up learning a lot about climate science and a lot about the economy and things like this. And it was really bizarre because I made some predictions and wrote this story. And then when I first showed it to some local science fiction authors who are sort of my readers, helping me get through the initial stages of the draft, one of them was like, "You are being way too extreme. There's no way it's going to happen this quickly." Now, there are a lot of things in the news, like for instance, Australia burning. I was like, "Oh yeah, the world will burn." And they were like, "No way." And I was like, "Oh my God, it's not supposed to happen for another 50 years. Why is it happening now?" So yeah, here's some examples of the art. So yeah, the idea is what happens to neuroscience, what happens to, sorry, I'm kind of, it covers a lot of topics. So right now there's this sort of push for interdisciplinary study, multidisciplinary STEM. People are like, oh, well, in the real world, when you're working on a project, it's usually not you are just thinking about mechanics and you are just thinking about biology and you are just thinking about the finances of the project. I mean, sometimes it is, but a lot of times now, people working on projects, they tend to have a little more overlap into each other's fields, right? And there's a little bit more sort of, everybody should know a little bit about a lot of different things in order to really be able to push innovative projects forward. And so, with that kind of mindset going forward, how will that develop? Given all the crazy advances in technology and given how quickly the internet has totally changed the way that we interact with each other, the way that we build societies, what will be the next few decades of this rate of rapid change? And then also, in my graphic novel, I predict that there's a horrible climate apocalypse and everything goes to hell and we have to kind of start over. So then when we are starting over from that starting place of, we reached the internet era, we reached the Internet of Things era, we reached the artificial intelligence, machine learning era, all of that broke down. So we have to start over, but where really is that starting over point? So that's kind of what I'm exploring with this. And then intermingled with, at the end of every chapter, is some neuroscience diagrams. So that people reading the graphic novel, it's like, oh, you like comics? Great. You like science fiction? Great. Here's also a little bit of neuroscience to learn. So yes. Oh, right. Okay. I have two last songs. The first one is a little depressing because it's about climate change. But then the next one gets happier, I promise. Okay, so. Jackets in June just ain't my kind of tune for a plump summer. Voraciously take the heat that it makes, decimate London's pace. They can eat our cake today. Don't wanna wait today. Won't be long till it's gone. So be merry with the They itch for your blood, they will make it flood into cold coffers. Procedural twine ensnares our minds, you're inclined to be blind to the other cheek today. Can't handle the weight of today. Won't be long till we're gone. Now we're wedded to our fate. Today's to look beyond the haze of money-making crazy the atmosphere. keep us, to bleed us, and feed the machine of won't be long till we're all gone. So get ready for our fate. So to end on a happy note, something that both features in the graphic novel as a theme and also something that I've learned over the course of my PhD is there have been so many moments where I was like, gosh, I really wish somebody had already figured out a way to automate this so that I could just send a Python script running and I wouldn't have to like watch these videos of cuttlefish for hours and hours again. But having watched the hours and hours of cuttlefish videos, I realized that there are some insights and things that I learned from doing it the slow way that I probably would have missed completely if I just told a computer to go do it. And so the last song that I have for you is called We Don't Need the Robots, But It's Okay to Love Them. We don't need the robots to feel for us. If someone else breathes, no oxygen fills my lungs. Remember to feed the organ that believes in the spirit of being kind. Hands and mind are mine to give to you. We don't need the robots to work for us. If someone else sweats, I have not gained your trust. Remember we bleed from underneath an armor pretending to save us time. Hands and mind are mine to give to you. Thank you to you for inviting me down, and thank you all for being here.
Thank you, Danbee. So I think we can take a few questions. Is there anyone have questions?
So, sorry, quite academic. Do you get the consent during the experiment in the aquarium?
So we tell them that we are video recording their eyes, but then because and then we replay the video for them. So the way that it happens is we watch the thing, we record it, and then we replay it so that they can watch it again. And while we were hosting, I kept asking people like, is it okay that like you can tell me that if you don't want it, I'll go in there and I'll delete it. And like people were like, yeah, no, this is great. Like and people were like, I don't think you can even tell that it's me. There were some people who were like taking selfies like with their video recording and stuff. So I think after a while we decided and we talked to the Sea Life Center. They were like, well, as long as it's clearly signposted and we're not collecting any identifiable information. And of course, this might change in the future, but given the current technology, because it's so zoomed in and the focal length is so small, it's like completely out of focus until they like literally come up here and then it's just eyeballs. So we kind of collectively made the call that it was okay to not get people's explicit consent because we're clearly signposting that we're recording their eyes, we're replaying it for them. And they're also in sort of like a closed environment where it's very easy to be like, hey, I need to talk to a Sea Life Center worker to let them know that I have a problem with this. And in the 11 months that the experiment was running, nobody said anything. So yeah, it's a little bit of a like, I'm not sure, but there was no precedent for this sort of thing. And every time we asked for advice, like even the UCL review, like ethics review board, they were like, well, I mean, you're clearly stating it. Are you collecting any personally identifiable data? And as soon as we said no, they were like, okay, then I don't know why you're asking us anything. So yeah, so I think it's okay. Yeah.
First of all, great talk.
Thank you.
I don't know if you're planning on going into academia, but I kind of hope you do because I think we need people with your energy. And, appreciation for knowledge translation in that space. Saying this as a fellow neuroscience PhD student. But
Thank
I have
you.
more of like a technical question for your first study. Did you figure out like what the adaptive kind of function was of that verb?
No, I think so. Basically, the paper that we're going to be putting out has an enormous discussion section where I'm like, here's the little like baby step of a thing that I found, which is how to numerically describe this effect, hopefully unambiguously in a way that is replicable. But now, so one of the biggest challenges with this analysis was that at the time of data collection and at the time that I was doing most of the data analysis, we could neither find nor build an algorithm that could reliably track a creature that is actively morphing visually. Now there are things like Deep Lab Cut, Deep Pose Kit, and those are just published very recently. So basically, I end the paper and this section of my thesis with a recommendation of someone should replicate this study and use Deep Lab Cut or Deep Pose Kit to track these animals throughout the entirety of the session. Because I did like 6-second clips around the tentacle shot. And that's why the plot that I showed you is only so long. But yeah, it would be great to kind of get more information on, like I took a baseline of like just before they shot the tentacles, and that's where I compared the edginess. But maybe their edginess is already increasing by that time from I'm just swimming around, And so like, yeah, further work should definitely be done on this. And I think in terms of trying to decide like what is the adaptive role of this thing, it definitely needs to be more work done.
Do you think, like, can you use machine learning to predict, like, to just look exclusively at the pattern to see if it
to predict if it will catch or not?
Yeah, I mean, like, could you draw any correlations between those two things?
Maybe, I don't know. So yeah, there's a couple of things there. One is that if the pattern is being controlled by the animal, like then the animals, like the animal would somehow have to know before. So we would have to.
I'm not thinking like that you use it to predict whether they'll catch it,
Oh,
or whether
okay.
you can see whether if you just look at that pattern, if that would align with them catching it. Do you know what I mean? If you just draw a correlation between
actually,
capture
okay.
and like pattern.
There's a version of a talk that I did about that. There's a YouTube video where I tell you everything that I've told you now, and then I just show you like a zoomed in like box of the cuttlefish back, and I ask you, do you think it's a catch or a miss? And it's actually like I think it's pretty. Actually, maybe I can, if there's internet here, we can just quickly go to YouTube.
I mean, it might also just be one of those things like that Darwin got frustrated with peacocks. You know, it may not have an adaptive function.
Yeah, maybe it's just like some selection gone wild into this weird corner. Yeah, so yeah, it's this beautiful, delightful mystery that like we don't actually. I think there would need to be a lot more work done to say either way, and then you're right, and we might just 30 years later just be like, well, no, we have no clue. There's no point. Yes.
Actually, kind of on the discussion, has anybody done any observation? And I imagine it probably be very difficult, but of cuttlefish stay in the wild, and once they've caught it and had this color group as to what happens next, if any other animal notices or responds or anything like that, or has that not been done?
So there hasn't been like super rigorous studies done, but the reason why we're pretty sure it's evolutionarily robust is because it shows up in a lot of different species. It shows up under all kinds of lighting conditions. It shows up in like laboratory settings and in the wild. It shows up for both like when they're catching shrimp versus when they're catching crabs versus when they're catching fish. So it based on like current anecdotal evidence, it seems incredibly robust. So like if it was something that was making the cuttlefish get caught and eaten more often, then you would assume that the genetics of having this weird color burp would never get passed on. It wouldn't continue.
It may have some unknown secondary function, perhaps, which doesn't
Maybe.
Well, so when they're mating, they put on this like crazy zebra stripe pattern, and they start to kind of typically they stop caring about hiding, and they're just like, I'm here, I'm ready for sex, I put on my sex colors, please, like, you know. So actually, a lot of times, what they'll do in the lab I worked with also, they'll keep the temperature low enough so that the animals don't sexually mature. Because once they sexually mature, they just put on their mating stripes, and they're like, I don't care about anything else. And they stop camouflaging as much. So yeah, I mean, yeah, it's sorry, I don't know if people actually want to give this a go, but there is a, oh God, if I add summary. Yeah, so my whole data set is actually online in low res, and if anyone, you know, has a machine learning algorithm that they want to test on a really challenging data set, I have the full data set online as well. Okay, this is quite long. You can find this later and you know, watch the whole video yourself. But let's go to the test. Okay, yeah. Okay. Yeah. So the way that I phrase the result here is, I should update it. Okay, so now this is the game. Can you guess? Okay, what do people think? Yes.
Nice.
Yeah, very good, very good. Okay. Woo! You all are experts now. What do you think? Well done. There you go. Yeah. Oh, and this is cute. This is when we're feeding them in the winter. I like decided to put on a GoPro. And so yeah, it's funny. Some of the animals here in their home tubs show the color burp, and some of them don't. This is also another thing to test more robustly. Like that guy, I don't know what's going on. Yeah. But yeah, they're really cute. They're like this big. And like if you wiggle your finger in there, actually, I realize, like, I understand now why all the stories of the Kraken and things show up, because like even though they're tiny, when they like rear back with all their arms and then you see like the beak, I was like, oh my God, I know you're tinier than me and I can squish you instantaneously, but I am deeply frightened.
Do they show dominance? Is there like a more dominant cuttlefish?
There is a hierarchy. It's more like the ones that are submissive just go hide, and it's a more passive aggressive kind of yeah. Oh, and also totally when they're in their home tubs, I've like held the thing above water, and then yeah, that one totally did the color burp. But I will hold shrimp above water, and they will peek their head above water in order to try and like. Sorry, there was a.
Can I be for communication? I mean, this changing pattern.
Maybe it's like, oh yeah, I totally caught a food.
I don't know about their social lives or I don't know.
Yeah, so we used to think that cephalopods were totally like isolated. They didn't have social lives at all. But there's been now, I think, 3 like cephalopod cities. Like the first one was called like Atlantis or something, and like it was built around like an old shipwreck, and so people were like, oh no, this is totally an anomaly because there was a human like thing that went down into the ocean, and then they built a community around it. But then they found another one. This is all around Australia. They found another one where, oh no, we're not going to watch the draft version. They found another one where it was completely entirely made by the animals. Like they brought in like rocks and shells, and then they created like little, like apartments, and then they have they have like a social hierarchy where they have squabbles over resources when things get too crowded. Like it's weirdly human. Like I'm not even kidding when I say like if we really mess things up on land, they're ready to take over the planet. They're just like, just you wait, fuck things up enough, we're gonna come take over. So again, like because they're marine animals and because we haven't paid that much attention to them until recently, there's a lot of unknowns. I mean, it's very exciting, right? It's like, do you want to come do science with me? Let's go scuba diving. Yeah. So yeah.
I have a question because you calculate the cuttlefish in water tank.
Mm-hmm.
Yeah, is it the behavior different from the natural? They were living natural, I mean, in the sea or the river?
Yeah. So that's definitely a possibility. But like I said, anecdotally, so just based on stories from marine biologists and scuba divers, this particular behavior, the post-tentacle shot, like color burp, this has been observed in the wild as well. Yeah. And also, so the interesting thing about cuttlefish is they're so delicate that it's really difficult to like raise them in the lab unless you make things as natural as possible. So like the videos that you saw of me like with them in their home tubs, that is water pumped directly in from the ocean. That's why they're so difficult to work with, because like rats and mice, they're really adaptable. Like you can put them in all kinds of like sterile, weird little boxes and they'll just like, okay, fine, I will adapt. But cuttlefish, if you make it too unnatural, they just die. So like sort of out of necessity, in order to study them even in the lab, you have to make things as natural as possible. You saw there were lots of like hiding places like fake ferns and little, and I showed, I kind of, and a fake fern. So the way that I decided that they were no longer stressed by the move from their home tank to the hunting box was if they settle somewhere near the home base area and they're not completely white has been shown through observation that this is a sign of stress for them. And so yeah, like you really have to, they're quite spoiled, but I mean, they just want to be in their natural environment. They're quite delicate. So it's really difficult to work with them too far from their natural setting. So sort of out of necessity, people have been forced to, the researchers have been forced to adapt to the animals, which I think is the correct way around. Yeah. But yeah, we definitely, study this robustly in all kinds of situations, which is why it would be great to figure out how to do this in the field, etc. Yeah.
Do you plan to include the songs in your defense?
Sorry?
Do you plan to include the songs in your defense?
I haven't decided yet. I'm trying to like get a feel for how much weirdness my thesis committee is going to be able to tolerate. Like they were all like graphic novel, what? And I'm like, okay, well, what if I play some music in my defense? Like, we'll see. I'll have to gauge it as we go. Yeah, but I hope you guys enjoyed it.
So, any more questions? Thank you. Thank you. Yeah, so there are some drinks there, so please feel free to take them. Yeah.